Good morning. It's my pleasure to introduce today's speaker. Dr. Craig Williams is a clinical professor of pharmacy at Oregon State University College of Pharmacy and associate professor of medicine at OHSU. His research is in the area of chronic disease management and prevention with a focus on cardiovascular risk reduction. And his clinical practice is with the Division of Family Medicine at OHSU, where he focuses on the management of chronic diseases and transitions of care. He's won multiple teaching awards for both didactic work in the classroom and patient care work on the hospital wards. And he gave this talk at one of the OHSU internal medicine updates, and it got really good kind of evaluations. And I thought, just in my world in diabetes care, there's so much going on with the FDA and the approval process, and I'm often kind of confused as to what's going on with that and get a lot of questions from patients in that regard. So I thought this would be a useful topic for us to hear about today. So thank you, Dr. Williams, for coming over and speaking to us.
Thank you, Liz. Hopefully that buzzing is not my microphone, but thank you very much for the invitation this morning and the introduction. It's truly a pleasure to come across the river and to speak to colleagues and peers here at Providence. So in the last few years, there's been a fairly robust exchange of some personnel between OHSU and Providence, so I feel like it's a bit of a home away from home, and some familiar faces here. So I will this morning talk a bit about, as Liz alluded to, the process of FDA approval. I think most of us know something about it. I knew something about it before we undertook kind of a project a few years ago with a few folks up at OHSU, and then I learned a lot more about it, which I may be a bit embarrassed to say I didn't know as much about as I probably should have as a kind of academic pharmacist for 15 years. So the subtitle here is somewhat appropriate, kind of what I've learned about the drug approval process. And as Liz alluded to, there has been a lot of excitement in the diabetes area lately, and new trials coming out and trials contradicting one another. I'll actually be on a conference call next week for Prescribers letter, and that will include Stephen Nissen, one of the co-authors of the recent LEADER trial. So lots of things happening in the diabetes world around FDA drug approval. But this morning we'll focus a bit more on the kind of broader aspects of kind of what the FDA does and what drugs go through before they get into our hands as prescribers and providers and even as potentially as patients. Then I will use to kind of exemplify that this project that we had worked on that was just published about this time last year in one of the JAMA journals. And it really highlighted for me some of the kind of pitfalls of the process and things I'm glad I now know about the FDA drug approval process. And I'm happy along the way to take questions, so if you have questions, please feel free to raise your hand. I try not to overload us with information and slides this morning. 8 o'clock on a Wednesday morning is not the best time to go through 100 slides, so we should be okay on time, and I'm happy to take questions during the talk. And I kind of consolidated the 3 objectives I sent on to kind of this one for the morning, which kind of summarizes them. And really something I kind of learned in the process of this project that we'll go through. But the main objective here is to understand why generally drug manufacturers know the most about a drug. It's particularly true when the drug is launched and in the first few years when it's available. The FDA knows something less than that, and then we as prescribers, clinicians, and patients know less than that. So it's a bit of a kind of a hierarchy to how the information kind of flows downstream. And that's an offshoot of the way the process is designed and the way drugs go through the FDA. So we'll talk a bit more broadly about that process and then talk a bit more specifics. Well, first just to highlight, and it's okay if you can't see the details there on the bottom of the slide, but you can probably see the blue line kind of dancing across the bottom there. This is a very nice interactive slide, as the New England called it, published back in June of 2014. I think it was in the June 26th New England Journal of Medicine. You can still go there online and pull it up. You can kind of click on different aspects of it, and it'll kind of give you information, so it's kind of a fun interactive slide. But a couple things to point out this morning, and then we'll go through a couple of the kind of key aspects here that kind of led to the birth of the FDA and how it's evolved. First, just to kind of point out down there at the bottom, that the timeline on the very bottom goes from 1940 out there to the left, all the way over to about 2014 to the right. So it's looking at about 6 decades of drug development and drug release here in the United States. On the y-axis there, it's at 0 at the bottom up to 60 on the top. And you can see that on average, the number of new molecular entities, or what become new drugs that get approved every year, is typically around 20 to 30 to as high as 40. We'll look a bit at that spike year, that 60 out there. That was 1996, and we'll look at that in a bit more detail in just a minute. But generally, the FDA is dealing with around 30 to 50 new molecular entities hitting the market every year. And each of those new drugs has its own story to kind of tell in terms of how it got through the FDA, the studies it underwent, how good those studies were, if those studies were published. Each drug kind of has its own story in getting through the FDA, and we'll look at kind of a couple of those specifically as we go along. But just to point out a couple of the kind of acts here that were important in the evolution of the FDA. As we know, the FDA was kind of born, the modern FDA was born in 1938. The FD&C Act, or the FDA and Cosmetic Act, was passed by Congress in 1938 that really created the modern precursor of the current FDA. And that was passed in response to the elixir sulfanilamide. And sulfanilamide was an antibiotic available for about a decade at the time. There was some consumer demand for a liquid form of the antibiotic, so we still like to respond to our patients and consumers today, and they did back in the 1930s as well. So a drug company made an elixir form of sulfanilamide. They unfortunately used basically a version of antifreeze, used diethylene glycol to kind of to suspend it in solution. It tasted pretty well, as you may hear from some patients today. It smelled pretty good. It was pretty palatable to patients. Unfortunately, some patients consumed it in enough quantity to cause renal failure, and there were about 100 deaths associated with the drug. And so this act had been kind of circling Congress for a year or two, and then this kind of tragedy pushed it forward and passed in 1938. And what that act established was you had to give the FDA 100 days, essentially, notice that you were going to bring a drug onto the market. And the FDA had those 180 days to determine if that drug was safe. If it wasn't safe, then it moved forward, but the FDA had that kind of time that could stop the drug if they found something unsafe. So that was 1938, FD&C Act. If we come forward a little more than a decade, 1951, the Durham-Humphrey Act establishes a need for prescription for certain high-risk drugs. And a lot of interesting history written around this. But before 1951, you pretty much walked into the druggist or the drugstore and bought your drug. You didn't need a prescription. Starting 1951, you needed a prescription for certain drugs. Other drugs you could still purchase. So the American Medical Association is very involved in that, and there's some very interesting history around that act. But 1951, need for prescription for certain high-risk drugs. In 1962 now, the FDA moved into the efficacy realm, so the Kefauver-Harris Act required proof of efficacy for approval for the drug. So late 1930s, 6 months to establish safety. 1962 now, need to show efficacy as well as safety for the drug to move forward. Just a couple of things I'll point out. You see there's kind of a lot of acts that occurred and a lot of modifications there in the 80s and 90s, but a couple of things you may be familiar with that I'll just highlight. In 1988, the Fast Track was established. This was largely in response to the AIDS crisis and HIV drugs moving very slowly through the pipeline, and given the high mortality associated with AIDS. In the 80s and 90s, there was a lot of patient activism for this act, and Fast Track does still exist today. So it's not entirely clear how a drug enters Fast Track, but it is there for certain drugs where there is an urgency for the drug to reach the market. And then I'll say a little bit more about this in a couple slides, but 1992, in the Clinton administration, PDUFA passed. It's still fairly politically a bit of a hot button. It was renewed in the Bush era, so both Democrats and Republicans have kind of had hands on it. But PDUFA established a system in which the drug manufacturers partly paid for the review of the drugs. So essentially established a user fee to submit a drug to the FDA. Again, remains somewhat controversial today, and I'll say a little bit more about that, but not a whole lot. But 1992, PDUFA passed fees for companies to submit drugs to the FDA. It has allowed the FDA to hire quite a few more reviewers, and it has expedited the drug review process, but does still raise some questions today. Well, let me now we'll focus a little bit on the 1996. So the 60 new molecular entities approved in 1996. It was the peak year for new drugs in the United States in the late 90s. And as I kind of said at the opening of the talk, every drug has its own story to tell and how it gets through the FDA. So the general approval process is kind of a broad process, but each drug goes through its own kind of act. Some drugs get through fairly quickly in just over a year. Some drugs get tied up for over a decade. So if we look at what came out in 1996, and depending on kind of what area you practice in, a lot of these drugs are familiar to you. I put them into a couple of categories, notable successes that you may agree or disagree with, depending on your practice and what drugs you like. Notable failures, just below that, and what I called also rans. I think if you took any of those years where drugs were being approved and you looked at what got approved that year, you could probably kind of say there were some drugs that were real advances, some drugs that kind of were definitely not real advances, and some drugs that were kind of somewhere in the middle. I'll just look at a couple of the successes that year. That was the year that atorvastatin was launched. So Lipitor came out 1996 as one of the new molecular entities. Olanzapine or Zyprexa, one of our carbapenem antibiotics, meropenem, kind of a new different class of antidepressant, mirtazapine. A couple of the failures, dexfenfluramine, there's a cousin of the fen-phen drugs that were later withdrawn, the weight loss drugs that were withdrawn for valvular defects. We had another quinolone, I think at the time the fourth quinolone to enter the U.S. market, sparfloxacin, which didn't last too long and got removed for QTc prolonging and arrhythmia risk. Got another leukotriene synthesis inhibitor, Zyflo, so a few drugs that really didn't go on to do anything and or got withdrawn from the market. And then a few what I'd call kind of also-rans, things we probably could do without, but not bad to have on the market. Another ACE, another ARB were both released that year, so Trandolapril and Valsartan. Both came out in 1996. Donepezil for Alzheimer's type dementia. Fexofenadine, Allegra, so a low stating antihistamine. Interestingly, Allegra, for those who may know, is a cousin of Seldane. Seldane or terfenadine got removed from the market because it had QT prolonging effects as well. And Allegra is actually the biometabolite of that drug, which doesn't have the heart side effects. So a lot of drugs that year. But clearly, I'd say we can probably agree the FDA does not always get it right. Not every new molecular entity that comes through the FDA process really is an advance in therapy or even always necessarily safe at the population level. So let's look briefly at kind of why the FDA doesn't always get it right. And this is certainly in the context of the fact that the FDA usually does get it right. And the FDA is working very hard on our behalf to get it right. So they are obviously a taxpayer-funded institution. They're doing a pretty good job, but they have a very difficult job to do. So the little figures here to the left, I took right off the FDA's webpage. And if you go to fda.gov and you kind of search drug approval process, you can find lots of interesting information. The figures don't look particularly sophisticated to me, but I figured my tax dollars paid to create them, so I'm going to use them here and not recreate them myself. And this is, the FDA lays out what they call a 7-step process for a drug coming to the U.S. market. And as I'll show you in the next page, I kind of inverted step 6 and step 7 because of the reality of how drug approval works. But here are the first kind of 5 steps. Steps 3, 4, and 5 are phase 1, phase 2, and phase 3 testing, which you guys have probably heard those terms before in training or in practice. I think we talk a lot with our students and residents about what phase 1, phase 2, and phase 3 are. Those are really kind of the 3rd, 4th, and 5th steps of the process. So just to kind of go through this briefly at kind of the broad level, step 1 is animal testing. So as I came over from OHSU this morning, there were a few more primate center protesters out in front of OHSU. So animal testing certainly remains a controversial area in 2016. But step 1 is testing drugs in animals first in a completely preclinical phase of drug development. So step 2 then is investigational new drug applications filed. And here, one of the things I learned in the process of going through our project with some of the folks I worked with, the company here often seeks the advice of the FDA in how to put together an investigational new drug package. So once the company feels like I've got a drug that looks safe and efficacious in my animal models, I want to think about moving this into the human area, they do often consult with the FDA. In fact, the FDA really wants to know what they plan to do, because if you plan to bring this drug to the U.S. market, you have to tell the FDA what trials you're going to do. The FDA has to be aware of those trials before they're conducted. So essentially you have to be registered with the FDA. I'll say a bit more about that in a couple of steps here. But it's interesting to me that there's quite a bit of consultation between the manufacturers and the FDA at this stage as the new drug application is being filed. So once that's in and accepted, phase 1 testing can begin. This and the numbers there to the left kind of show how many patients get exposed to the drug at different levels of testing. So in phase 1 testing, which is the 3rd step of the process, typically very small numbers of patients, so less than 100 patients involved in phase 1 testing. This is healthy volunteers. These studies often pay quite well. I was chatting with a colleague that I knew from Indiana before coming back out here to Portland, and our offices, when I worked at IUPOI in Indianapolis, we moved into the top floor of the county hospital, which had been occupied by Eli Lilly and had been their clinical drug testing center. So patients were housed there for often days or even a couple weeks at a time, reimbursed pretty well, often to take drugs in phase 1 studies. So drugs where we knew they looked fairly safe in animals but didn't know what they looked like in humans. So in phase 1 testing, healthy volunteers exposed. Companies looking for obvious toxicities, things that should stop the drug from moving forward. This is where they determine the kinetics of the drug, how's it cleared, what happens potentially in patients with kidney failure or liver failure. And they look at doses. What kind of doses appear to have the effects we may want for this drug? If you survive phase 1 testing, you go on to phase 2 testing, and now we get a few more patients involved. So typically in phase 2, 100 to 500 patients exposed. This is now looking for some efficacy in the population you want to use the drug in. So in phase 2 testing, if it's a cholesterol drug, we're giving it to people with elevated cholesterol or with heart disease, and we're looking for some effects. Often here with surrogate endpoints. So kind of back to the diabetes debate, for a long time diabetes drugs were approved based on A1C and glycemic control. And as many of you may now know, to keep a drug on the market, you've got to do cardiovascular safety testing for a diabetes drug. But typically surrogates used here in phase 2 testing. Again, relatively small numbers of patients. So if you survive phase 2, step 4, you move on to phase 3 testing. Now typically a couple thousand patients exposed to your drug. And this again, we're looking for effects in the target population in enough numbers to establish some effect size. So now we really want to know, what does this drug do in our target population? And here again, a lot of FDA kind of consulting involved. So as the companies design phase 3 trials, and it's a phase 3 trial that typically gets the drug approved or sinks the drug. If you get to this point, it's really critical that phase 3 trial goes well for the new molecular entity. At this point, if you want to use that trial to get the drug on the US market or to change the labeling of that drug, the FDA must know about that trial before you do it. And the idea there is pretty simple. The FDA doesn't want you going away to some dark corner somewhere, doing a big trial of 5,000 people and bringing it to the FDA and saying, here, look at our great results. The FDA wants to know what you're doing, where you're doing it. They need to have a chance to audit your files as you're going along. So a lot of consultation between the FDA and the manufacturer at this step. Nothing necessarily public. This is all privately done. This is typically in this country the drug manufacturer. If the drug does not get approved, that data does not have to be publicly released. So if a big trial was done, drug failed, never comes to the market, that remains proprietary data with the company. There's no obligation for them to release that data to us, the prescribing or consuming public. So phase 3, a lot of FDA consultation, a couple thousand patients involved. And then the last steps here, on the FDA's website, they have phase 6, or they have step 6 before step 7. I tell you, in the real world, it's more like step 7 first and then step 6. So if you do well in phase 3 testing and your trial looks good, you move forward with your new drug application. So you apply for this drug to be a new molecular entity in the United States. What the FDA says is after phase 3 is phase 4, and phase 4 is where the company's supposed to design their post-marketing surveillance. So the company tells the FDA what they're going to do to monitor ongoing safety and efficacy of this drug once it's out there in the hands of us, the prescribing public. In the reality, though, there's not much teeth behind phase 4 testing. The companies do put something together, but it's often not followed up on very well. So in reality, once phase 3 is done successfully, the new drug application can move forward. And once the drug's on the market, some degree of phase 4 surveillance does begin. But I'd say it's more in the order of new drug on the market than phase 4 monitoring, rather than vice versa. All right, so it's a lot of steps. So as I said at the beginning, things often go right. A lot of drugs move through the FDA approval process just fine. They are significant advances for us. Truly, pharma does do a lot of great things. We wouldn't have the drugs we have without them. But occasionally things do go wrong. So as far as things to kind of be aware of as prescribers and things I learned, we'll talk about a couple kind of key points here. Step 1 is just be aware that relatively few patients are exposed to that drug before it comes on the market. So you probably all have colleagues that you've heard say, I'm not going to be the first clinician to prescribe this drug when it comes out. I want it to be out for a while, make sure nothing kind of bad happens. So someone's got to go first. Someone has to be that brave initial prescriber and use the drug. Oftentimes the drug is fairly well vetted through phase 3, but sometimes not as well as we might like. So just know there are relatively few patients exposed before the drug hits the market. The implication is if you have a side effect with a frequency of certainly less than 1 in 500, you're definitely not going to detect that through phase 3 testing. There's not enough patients exposed to drugs through this FDA process to pick up a side effect that has an incidence of less than 1 in 500. If you're in the range of less than 1 in 100 side effects, you're probably not going to pick that up either. If you're looking at randomized trials of kind of 1,000 to 2,000 patients in one arm and 1,000, 2,000 patients in the placebo arm, it's going to be hard to pick up a side effect that might occur just a dozen or 2 dozen times in that population. So again, it takes a fairly robust frequency of a significant side effect to pick it up through phase 3 testing. And I'll show just kind of one slide on that in just a minute. The PDUFA Act, which I won't say anything more about in detail this morning, but just to kind of put up here again, happy to talk about in the question and answer session if you guys would like. But PDUFA remains an area of kind of controversy for consumer activist groups, for a lot of clinician researchers who disagree with it. The FDA re-reviews it periodically. Again, it's kind of gone through both Republican and Democratic Congresses and been renewed. But the reality is the FDA has a big job. It could use more reviewers. It's a taxpayer-funded entity. It's generally underfunded by us, the taxpayers. And this is a way to get more resources for the FDA to hire more reviewers to move along the process more expeditiously. So the FDA likes it. The FDA feels it's well managed and doesn't create any conflicts of interest. Bob Califf became the FDA commissioner just earlier this year, and he's a cardiologist researcher by training. All indications are that he likes the PDUFA Act as well, and they like having more personnel at FDA to do their job. And the reality is this pays for a lot of that. But it is essentially a user fee act on the drug companies whose drugs are pending before the FDA. And that does still create some controversy and debates, even among some of our congressmen. So as a question here is posed there, are the foxes in charge of the hen house? I think not. I think PDUFA's got enough kind of structure around it that it's probably a good thing, but I can certainly see the controversy around it as well. So I won't say any more about that this morning. And the third thing we'll touch base on is kind of that main objective. The manufacturer knows most about a drug, FDA knows almost as much, and we know less than that, and we'll talk a bit more about that. And then we'll close with a few thoughts on the fact that generally the FDA is a little concerned with the magnitude of effect. And that's pretty obvious when you go through some of these FDA review packets. What you need to get a drug on the market is a p-value less than 0.05 in your phase 3 trial. And generally, for lots of drugs, you need to beat placebo. So if you're better than placebo, enough better to get a p-value less than 0.05, you're in pretty good shape for getting on the market. That doesn't tell us as prescribers much about what is the absolute benefit of that drug. And how many patients do I need to treat for this drug to be effective? Do I have a number to treat of 10 or 100 or 1,000? And the FDA doesn't much care about that right now either. The FDA cares, are you better than placebo, not how much better are you or how good are you. So the FDA really is very qualitatively oriented towards the data, not particularly quantitatively oriented. And there's more people asking for that to be kind of changed, and for the next evolution of the FDA to be kind of from safety to efficacy to really quantity of efficacy. How good is this drug really? To give us more information on that. And we'll say a little bit about that at the end. So to come back to point number one, just kind of briefly. And a lot of you here may be familiar with this controversy when it came out, but it's a nice example of kind of the potential pitfalls of the relatively low exposure rate of patients in trials. The CETP inhibitors were developed, they've been developed by a few companies, most notably early on Pfizer about a decade ago. In 2004, a phase 2 trial was published in the New England Journal of Medicine. And if there's ever a phase 2 trial published in the New England Journal of Medicine, it's probably something pretty significant, because these typically are very small numbers of patients. And New England, as we know, is a pretty good journal to get your data in. So 2004, this paper was Effects of an Inhibitor of CETP on HDL Cholesterol. The numbers of patients here, you can see in the method section, 19 subjects involved, exposed to this new drug, torcetrapib. At 120 milligrams, it raised HDL by about 60%. So significant rise in HDL, 19 subjects. A lot of excitement around this drug. And then an unusually large phase 3 trial done. So at the time, the FDA told Pfizer, you cannot move this drug forward based simply on HDL data. They felt we didn't know enough about HDL cholesterol to approve a drug based on HDL. You can't get a drug approved on LDL lowering. You cannot get a drug approved on HDL raising. So the LDL is accepted as a surrogate, HDL is not. So the FDA told Pfizer, you need to go do a phase 3 trial. Pfizer did that phase 3 trial in New England Journal of Medicine in 2007. One of those two trials was published. You can see there the results on 15,000 patients, so an unusually large phase 3 trial. They're typically in the range of 2,000 or 3,000 patients, but 15,000 patients because they needed outcomes data. They needed heart attacks and strokes and cardiovascular deaths. And see there that all the confidence intervals went the wrong way, and some of them significantly. And in fact, the increase in death from any cause, which is on the bottom line there, was 58%. And this was in a little over 2 years, or about 2 years in this trial. So these patients that receive this HDL raising drug would have really been better off probably starting to smoke about 2 packs a day than be on this particular drug, which increased mortality by 58% in about 2 years. So it really highlighted the fact that we need pretty robust phase 3 testing, particularly for drugs that have surrogate data that we may not understand particularly well. And 15,000 is an unusually large trial. So if you cut that trial by 2/3, if you make it 5,000 patients, probably none of those p-values would have been significant. And the drug may have come to market based on the fact that it raised HDL but didn't lower events, and we might have figured this out 2 or 3 years later when a couple 100,000 patients have been exposed. So, but again, it kind of highlighted just reminded us that it is a fairly small number of patients that get the drug before it comes on the market. Well, let me say a bit then about kind of the main objective, that the manufacturers know most about the drugs, and we all know something less than that. So the project that I had worked on starting about 5 years ago that got published last year was clinical reporting bias in clinical trials looking at second-generation antidepressants in the treatment of anxiety disorders. As I kind of said in the opening, I should have known more about the FDA process, and I should have known more of this stuff. I blame my educators at pharmacy school and residency that I wasn't better educated in this process. But I had to learn some of this as I was going along. And fortunately, the kind of senior person on this was Eric Turner, who's the last author here. And Eric, who some of you may know, is a psychiatrist up at the VA here in Portland. And he was an FDA reviewer before he kind of left the FDA and came back into academia. And that seems to bring a lot of wisdom with it, having spent some time at the FDA, then come back into academia. So I learned a lot in the process that I can now share with colleagues. We certainly weren't the first. Turns out lots of people certainly knew a lot of this before we did, and I did. So if this talk this morning kind of sparks some interest and you want some good reading on the topics that we'll kind of cover in the kind of remaining half hour here, this is a very good editorial published by Schwartz and Woloshin, who are a husband-wife combination. This is in the New England Journal on October 29th, 2009. The title was Loss in Transmission, FDA Drug Information That Never Reaches Clinicians. So kind of alluding to that fact that the manufacturer knows the most about a drug, the FDA knows something more than that, and we know less than that. So one of the things that I learned in kind of doing this project was that first quote there from the article. Drug labels are written by drug companies, then negotiated and approved by the FDA. So probably if all of us in this room got together and said, let's design a process for drug approval, it probably would not involve the manufacturer writing the label, then being approved by the FDA. It might involve an oversight committee with clinicians writing that label. It might involve the FDA writing that label, but it probably would not be the manufacturer. But as a result of that, Schwartz and Woloshin kind of maintain, and I'll show you some data and we can decide ourselves at the end. But as a result of that, they say much critical information that the FDA has at the time of approval may not make its way into the label and or into the relevant journal articles. So the FDA has no control over what the company publishes based on the research. And the FDA does have control over the label, but as I'll show you, sometimes it doesn't seem like they do. The label may not really reflect what we as clinicians may want to know about that drug. But again, it's kind of a consequence of the process that's put in place. Just to share briefly, I did send the slides on, so the copies of the slides are in you guys' possession. If you have motivated students and residents here looking for projects, you can really find a lot of great data at the FDA's website. So I put in a few slides here kind of showing how this data can be found. And again, this is our tax dollars paying for this process. If you go to fda.gov, you can click on that drugs tab up in the upper left there. Excuse me. And you get to this page. Over on the right there in the middle, that says drug information, drugs at FDA is that hot link over on the right. If you click on drug information there, you'll get to the screen where you can type in a drug of your choice. And type in the brand name here. If you want to get some more information on this drug, it's all filed under the brand name and the manufacturer that brought the proprietary drug onto the U.S. market. So type in the brand name of the drug. I'll kind of show an example from our kind of project that involved Zoloft. So type Zoloft in there. You get to the kind of Zoloft page, and if you click on that, you can then type on the kind of dosage form, and either one should work. So if you click on the Zoloft tablets, you get to this page. It has some very interesting information added. And this hot link that says approval history, letters, reviewers, and related documents. I know you can't see that too well from the back of the room, but again, if you kind of just go through that process at fda.gov, you'll get to this section. And that's where the FDA puts much of its review. So we spent a lot of the summer of 2011 and 2012 going through these 300, 400-page PDF documents, which unfortunately are not searchable. They're not nice PDFs like you get from journals. They're just kind of a scanned PDF, so it's just a lot of hand kind of work and highlighting. And you can get a lot of interesting stuff there on what happened to this drug as it moved through the FDA. And I'll come back to that in just a minute, but to kind of divert for a second, I see at the top, FDA.gov, you may get famous. When Steve Nissen published his kind of still controversial data on rosiglitazone back in the late 2000s, this is where they got some of their information from, was from the FDA.gov website. So you need to get your hands on stuff that's not necessarily in print, because this doesn't all get published. So in the methods section there, Steve describes in the top line, they went to the website for the FDA to get some of the information that they use for their rosiglitazone meta-analysis. The 5 trials they found at FDA.gov that they would not have found otherwise are there in the bottom. So you see in the kind of bottom middle, about 2,000 of their patients that they had for this meta-analysis came from these 5 trials that they got from FDA.gov. And as he kind of describes in the methods section, they were quote-unquote archived at the website, and he gives reference 6, which is this website at the FDA for the Center for Drug Evaluation and Research. So they're accessing these data packets at FDA.gov, which has often unpublished information in it, and a lot of the exchanges between the FDA and the drug manufacturer as the drug is being approved. So again, depending on the topic you get interested in and look at, you may get a nice publication in New England Journal of Medicine, and then certainly we'll happy to have you in academia, and we like people that publish in New England. And Steve has done it fairly successfully, and not on only one occasion, using information that's available at the FDA that's not available at other places. So let me show you briefly then what we did with some of that. So I come back to this section, this approval history, letters, reviews, and documents at FDA.gov. And we're in the Zoloft section here. And here is this big messy thing you then enter. When you click on that, you get a bunch of hot links for this drug at different times, depending on what happened with that drug. And as I said at the beginning, every drug that goes to the FDA has its own story to tell. So we're kind of painting some broad brushes here this morning. And we'll look at SSRIs as kind of an example of the process. But every drug has its own story to tell. So here I circled in red there about a 400-page review from 1997 from July, and it says it was for a new or modified indication. So I know you can't read the dates, but down there at the bottom, Zoloft was approved 1991 for depression. Then 1997, the Pfizer filed an amended document to basically get a new label. And the indication they were going for was for panic disorder for Zoloft. So July 8th, 1997, Pfizer applied for an indication for panic disorder for Zoloft, and this is that review. It was, again, about a 400-page review. In the interest of time, we won't go through the 400 pages, but I'm going to highlight for you, show you a couple things that were intriguing. And I'll start with this. This is the overall opinion of the medical reviewer who went through all this data from Pfizer for Zoloft, going for an indication for panic disorder. And I think it's a sentence that I couldn't possibly do justice to without reading it, so I'm just going to briefly read it here for you, and then we'll see what we think about it. So this is, again, the medical reviewer's overall opinion of Zoloft for panic disorder. And he wrote in the end, the overall statistical and numerical superiority of sertraline to placebo is statistically marginally acceptable as providing some evidence, though not strong in view of the lack of robustness, in favor of the efficacy of sertraline in the treatment of panic disorder. And he says a 100 milligram dose showed overall better results than those shown by 50 and 200 milligrams. So I think if I showed this sentence to a room full of my residents up at OHSU and said, does sertraline work for panic disorder? I don't know what the answer would be. I'm not sure what this sentence is really conveying in terms of does sertraline work for panic disorder or not. There's a certain lot of hedging of bets there. He's saying there is some numerical superiority of sertraline compared to placebo. It's marginally acceptable statistically to provide some evidence, though not strong in view of the lack of robustness. So I've never used the word robustness, I don't think, in my career. I'm not sure exactly the implication is there. So I'm going to back up kind of one step from that and just show you. So the reviewer is looking at 4 studies for Zoloft for panic disorder. And so there's kind of that same sentence here occurs at the bottom of the highlighted area. But at the top of this highlighted area, he kind of talks briefly about the 4 studies that they were aware of, 3 of which had been filed with the FDA. So 3 studies, Pfizer said, here's 3 trials we're going to do. You're now aware of them, FDA. We're going to do them, and we'll look at them together. And the reviewer says statistically, for study 629, it showed reasonable statistical evidence. For study 630, it showed moderate statistical evidence. For study 529, it showed minimal statistical evidence. And then for 100, as based on ratios, for study 514, which was not reviewed, showed almost no statistical evidence for the efficacy of sertraline. So the trials were a bit of a mixed bag. among these 4 trials. And that 4 study number 514 wasn't reviewed because it wasn't pre-registered by Pfizer at the FDA for reasons that aren't real clear. The FDA was aware of it, but it wasn't filed with the FDA, so it wasn't considered part of the formal application for approval. So but it's interesting to kind of look at, when you read through these documents, you really get a sense of the nuance of these studies. So does this drug work? How well does it work? What happened in different trials? Was it consistent across trials? Was the drug always effective when it was studied? Was it inconsistent in different trials? You do get a sense of kind of the richness of the debate between the FDA and the manufacturer for these individual trials. But if you go to the package insert for Zoloft, for panic disorder, it does have the indication for panic disorder, as many of you may be aware of. If you look at the bottom of the slide here from the section describing clinical trials, it says the effectiveness of Zoloft in the treatment of panic disorder was demonstrated in 3 double-blind, placebo-controlled studies of adult outpatients who had a diagnosis of panic disorder. And that's it. There's no real discussion of the kind of debates around the different trials. There's no discussion of the magnitude of benefit, how the drug compared to placebo. Was the drug consistently effective across multiple trials? It just says in 3 trials it was effective. Now maybe for a lot of us in this room, we might think that's fine. The 3 trials were done, it worked, that's all I need to know in the labeling. But other clinicians may feel like, it'd be a lot more helpful to me if I knew there was some discrepancy across the trials and what the results were, and how much it beat placebo by, what was the placebo response rate versus what was the rate with Zoloft. So just to kind of show another example, because we looked across multiple antidepressants. This is venlafaxine XR, so Effexor XR. It similarly had 4 studies for panic disorder. You see down there at the bottom, the conclusion of the FDA was among 4 short-term studies, the results of 2 demonstrate the efficacy of venlafaxine ER in the short-term treatment of panic disorder in adult outpatients. So among the 4 trials that were done, the FDA says 2 were effective. I put it at the top, study 398 is one of those effective studies. And then study 391 in the middle was an ineffective study. So 4 studies done with venlafaxine XR, 2 were positive and 2 were negative. The one on top was positive. You can see the numbers of patients there in the middle column. And then the number of patients who were panic attack free, the percent free of panic attacks, which was a primary outcome in these studies. And it went from 34% with placebo to 54% with the lower dose to 61% with the higher dose of venlafaxine. So nice kind of dose proportional effect for venlafaxine. But then interestingly, in the middle trial, which had a similar number of patients, 168 on placebo, 160 on venlafaxine, and this was a dose ranging study, so multiple doses involved. The number of patients who were responders was 53% for placebo and 55% for venlafaxine, so no difference between placebo and venlafaxine in that middle trial. And the reviewer says in 2 studies it worked and in 2 studies it didn't. If you guys had to guess, does venlafaxine have the indication for panic disorder in the United States? It does. And if you look at the label, what we know from these in the label, for panic disorder in that section of the package insert, it basically says the efficacy was established in 2 double-blind, 12-week multicenter trials in adult outpatients meeting criteria. It does not say anything about the 2 trials that were negative. It does not comment on the magnitude of benefit of venlafaxine. It just says it worked in 2 trials, which is a somewhat arbitrary threshold the FDA has set for these drugs in this condition. If you can show them 2 positive studies, you got approved. But again, no real discussion of the kind of, I think, richness of what you get out of the reviews when you look at, how well does this drug work? Does it work well across multiple studies? So let me kind of, before we get to the last kind of topic of the morning, divert a bit and tell you a little bit about kind of our project, and which really came about from the fact that I think many of us, certainly in academia, would think, it's okay if the package insert's not too well written and doesn't have the information I want, because I don't use a package insert in my practice. We think we're too sophisticated for that, certainly in academia, that, we go out to literature and we figure out what does literature say works with this drug. That's fine to know what's in the package insert, but that probably shouldn't be what we're going to as our primary source for drugs we use in patients. So as I say, who uses a PI? We're too savvy. We'll go out and do a comprehensive literature search, or more appropriately, we'll make our residents do a comprehensive literature search, summarize it for us, and tell us what they think about it. So let me kind of divert for just a second on this question of, do antidepressants work for depression? And this is kind of some of Eric's seminal work he did that was published in New England just a few years ago, which a few of you may be aware of. So I'll go through it fairly quickly, but if we kind of back up a step with antidepressants, do they work for depression in general? So Eric's paper was published in January of 2008. Title was Selective Publication of antidepressant trials and its influence and apparent efficacy. See that to the left, they found 74 trials that were available to them, and kind of going to the FDA and at the FDA level saying, how many trials do we have of antidepressants for depression? There were 74. Of those, as the bar graph kind of shows, that upper bar on that bar graph is of those studies that were positive according to the FDA. So of the studies that the FDA said were positive, 37 of 38 of those were published. So an apparent efficacy rate of 97% in the published literature. At the bottom there, the trials that the FDA said were negative, that lower bar, 16 of those trials were not published, which is 67% of them. So 67% of the trials that the FDA said this drug was not effective were not published. 97% of those that the FDA said were positive were published. In the middle, there were a few questionable ones, none of which were published if it was negative. So for the questionable trials, that kind of lighter blue bar there is published but in conflicts with the FDA decision. So interestingly, if you go to the FDA review packets and look at those trials and read what the FDA says, then you find the exact paper in the published literature. It's often, as we say, spun by the kind of sponsor. So the trial will look positive when it's published, because they'll often use secondary endpoints, or they use kind of mixed matches of subgroups of patients. They won't publish the primary efficacy results, which the FDA said were negative. So the overall effect was, if we'd done a complete literature search for antidepressants for depression, we would have found, based on literature, of 51 published trials, 37 would have been positive and 3 negative, and then some questionable. So it would have had a very positive view on antidepressants for depression based on the published literature. Or, as Eric likes to display it, and these are Eric's slides, these next two, here's the journal view. So here's what our residents would have come back with after their complete literature search. They would have said 37 trials are positive, 3 were negative, looks great. But here's a view of that same data from the FDA's point of view. So much more mixed bag if you take all the data the FDA had and kind of put it together. So about even for whether or not antidepressants work for depression. So a big difference. Briefly, what did we find? What we found was not quite as sexy as that, so it did not get into New England Journal of Medicine, but was published in JAMA Psychiatry about a year ago. So we looked at the same drugs, but now for the indications of the anxiety disorders as opposed to depression. What we found there, what I kind of underlined in red, the 57 trials that we were able to find, 41 of those 57 were positive according to the FDA. So 72% of the trials that the FDA knew about were positive. When we found the published versions of those articles, 45 of those were positive. So 96% appeared positive in literature, 72% were positive according to the FDA. If you look kind of down there towards the bottom, if you take the published view of antidepressants for anxiety versus the FDA view of antidepressants for anxiety, you get about a 15% inflation in the relative effect size. So according to the published literature, they're about 15% more effective than according to the FDA. And look at the results from the two. So interesting. Not quite as strong of a kind of bias as we saw for antidepressants for depression, but definitely a publication bias going on there. And the fact that this exists kind of in the U.S. is somewhat a consequence of our approval process. Again, the manufacturers let the FDA know about the trials, but they're under no obligation to publish those trials once they're complete. So we get a mixed sense of them from the literature. And as we probably all have some responsibility in this to an extent as users of these drugs, we certainly can work on demanding more and better information from the FDA. We also kind of know from literature that when trials are negative, they tend not to get published for lots of reasons. So this is not necessarily the fault of the FDA. It's not even the fault of the manufacturers. So I review articles for journals, and when an article is negative, it's just less likely to get into publication. Journal publishers don't like negative studies. Reviewers often don't like negative studies. The investigators themselves may not have submitted the trial for publication if it was negative. So for lots of reasons, though, we do get, I think, a fairly strong publication bias in this country because the FDA knows about these studies, but the FDA doesn't publish them. They often don't even make them publicly available, especially if the drug is not approved. We're seeing this change a bit, so people are becoming aware of this. The studies that are negative are becoming more commonly published. Journals are aware of this, the importance of negative trials getting out there to us, the prescribing and consuming public of drugs. And then pre-registration at clinicaltrials.gov has helped to an extent. The fact that now is a public database where trials are being registered before they're conducted that we can go back and look to after the fact. So it's becoming more difficult for trials to kind of go under the radar and not be seen. So let me kind of wrap up then with a few comments on that kind of last point. That is it time for the FDA to kind of evolve again into the area of kind of more quantitative evaluation rather than just qualitative? Should the FDA be telling us a bit more about the magnitude of benefit and not just saying this drug works compared to placebo? And there's lots of opinions on this, and I'll share mine with you, and I'd be happy to hear what you guys think. This would definitely involve an evolution of the FDA, and this has been being editorialized a bit more about in the last few years. So here's an editorial published last year in JAMA. The title was Obstacles of Developing Cost-Lowering Health Technology. And one of the points that the authors point out in terms of the difficulty of kind of bringing cost effectiveness into the literature and into the approval process is the FDA itself. So as they say, getting a more qualitative review of drugs by the FDA would require expanding the FDA's mission beyond safety and efficacy to address costs. And this remains controversial, whether or not the FDA should be commenting on and getting involved in the cost debates. But as you guys know, drugs are getting expensive, sometimes very arbitrarily expensive, and that's not a process now that involves the FDA to any extent. The FDA in 1938 was established to review safety. In 1960, it expanded to look at efficacy, but the FDA does not care about what the cost effectiveness of a drug is. That's up to us as clinicians, to guideline authors. So it would require a lot of change at the FDA to make that happen. So just to point out and to kind of finish up on a point, when I showed some of the same data to kind of a grand rounds audience in family medicine up at OHSU about a year ago, there are very mixed opinions on kind of, should this kind of data be in the package insert? What should we be learning from the FDA about these drugs? So back to our kind of Zoloft example for anxiety. This is from that same review packet. This is Zoloft for panic attack disorder. And this is from that trial that interestingly found a greater effect from 100 milligrams than it found for 50 or 200 milligrams. Which to me as a clinical pharmacologist, that's kind of weird. If a drug works, to work less at a lower dose is kind of odd. Now there may be reasons for that, but that's kind of an odd finding. So was that a question or no? So to kind of, all the numbers here aren't in huge detail, but I think I, as a potential prescriber and or user of these drugs, might like to know quantitatively how well does this drug work. Not just there were 2 studies and it worked better than placebo, but how well did this drug work? How well did placebo work in these trials? So this is the data. The left-hand column there is 50 milligrams. The middle column is 100 milligrams. And then 200 milligrams is next to that. And then placebo is that last column. And this is number of panic attacks per week. And we're looking at the drop in panic attacks as the patients are on Zoloft to different doses for 12 weeks of the trial. And down there at the bottom in red, I've highlighted the percent change in panic attacks from kind of week 1 to week 12 of the trial. And then at the very end there are the statistical comparisons. Those are the dose compared to placebo at the end. You see individually, not too many significant values, but tell you cumulatively, when you put them all together, the 100 milligram looked really good. The 50 and 200 milligram didn't look so great. But look at the red numbers at bottom. This is a percent reduction in panic attacks by 50, 100, 200 milligrams, and placebo. So placebo lowered panic attacks by 48% in this particular trial, compared to 45% for Zoloft 50, 84% for Zoloft 100, and 60% for Zoloft 200. Now again, it's a little bit odd for the lower dose to work more effectively than the higher dose in clinical trials. We could postulate reasons why that may be. Maybe some off-target side effects come up at higher doses that diminish some of the benefit. But generally speaking, higher doses of drugs do more things for what we use them for. But I'll point out before we leave the slide, if you look at that top there, that baseline mean row. So at the beginning in this trial, of about 40 patients in each arm of the study, there was an average of about 7 panic attacks per week in the 50 milligram group, 17 panic attacks per week in the 100 milligram group, and 7 attacks per week in the 200 milligram group. So somehow in the process of this trial, we ended up with the middle dose ended up with patients who were having more than twice as many panic attacks as the lower dose and higher dose group. So a very uneven distribution at baseline. So not surprisingly, the group who kind of had more kind of panic attacks and panic disorder at the beginning got more benefit from the drug, even though it was a lower strength of the drug. So it turns out the drug does work, and tell you, and kind of in sum, the drug does work for panic attacks, but it's not tremendously impressive, and it's partly not so impressive because that placebo response rate is pretty common in that range of 40 to 50%. When we look at the depression trials, look at the anxiety trials, there's a very robust placebo response in these trials, which makes it hard for the drug to be a whole lot more effective. So whether or not this data should be in the package insert is still debatable. Should we know the placebo response rate was 48%, and here's what the response rate was for these other strengths? Would that be helpful? And I can tell you that among our family medicine colleagues up at OHSU, some thought, yeah, that should be in there. And they were a little bit angry that, I don't want to just know that 2 trials showed it was positive. I want this data. I want to know how well does this drug work. But other prescribers in the room, particularly for SSRIs, said, you know what, I always thought these drugs were not much better than placebo, and I mostly use them for the placebo effect anyway. I give them the patient and say, I think this will work. I think you'll feel better. They go out and they feel better. And whether or not that's a placebo response, the drug response, they didn't particularly care. But I think if you're a data-oriented clinician and you like to kind of have a sense of, how well does this work, I think it'd be nice to have that data somewhere in the public realm. We shouldn't have to go through a 400-page PDF document in the FDA to get this kind of data, in my opinion. So again, in the panic disorder label, nothing there about kind of quantitatively how well these drugs work, how they compare to placebo. So I will end with this is an image from that 2009 editorial from New England by Schwartz and Wolstein. They used a similar example. I'll just kind of show this at the end. So this is using the labeling for, in this case, Lunesta, which is one of the non-benzo sleep aids that are available in this country. And I think that's the one that had the glowing moth as its kind of a spokesperson in the television ads. But Schwartz and Wolstein here kind of give a quote from the label, and then they dug through the FDA documents and found the data, and that's represented in the table just below that. But the very last line of that quote up there says, Lunesta was superior to placebo on subjective measures of sleep latency, total sleep time, and WASO, which is wake time after sleep onset. So in the label for Lunesta, it just says the drug works compared to placebo for those 3 outcomes. But Schwartz and Wolstein went to the FDA document, pulled the actual data from that, and it's in that table down there below. So for how fast patients fall asleep, it was 15 minutes faster. So on placebo, they fell asleep in 45 minutes. On Lunesta, they fell asleep in 30 minutes. For sleeping, total sleep time, it was a 37-minute difference, 5 hours and 45 minutes compared to 6 hours and 22 minutes. And then for wake after sleep time, it was a 9-minute difference there in the bottom line. 30 minutes for placebo, 21 minutes of wake after sleep time for Lunesta. So I think they do a nice job in their editorial, not basically saying this is kind of bad or wrong, but just we should have that information. We shouldn't just be told the drug works. We should be told how well it works, particularly compared to placebo. That's important for us to understand, not just that the drug works, but how well it works. Because we're deciding whether or not we should use this drug in patients, and all drugs have side effects. And as we try to decide the risk-benefit ratio in our patients, we need to understand the magnitude of benefit to understand how much risk we should tolerate. So there's a lot of robust debate going on now in the kind of pharmacologic literature around how should labels be changed, how should the FDA communicate what they know to us as prescribers. So I'd say in kind of 2016, things that are definitely kind of good to know about the drug approval process. Drugs do still have relatively few numbers of patient years of exposure when they come onto the market. So if you're in that mindset, you don't want to be the first clinician on the block to use a drug when it comes out. That's maybe not such a bad mindset, but again, someone does have to start using it first for us to get that data. But again, not a lot of exposure when drugs launch. Approval may still be based on surrogate endpoints. That's kind of shifting a bit, but it's not always hard clinical outcomes that drives drug approval still in 2016. Labeling for drugs is written by the manufacturers and then kind of negotiated and approved by the FDA. So I won't say that's inherently necessarily bad, but something good to be aware of as prescribers. And then sponsors decide what trials are submitted for publication. So trials used in labeling, the FDA knows about, but what gets published is not up to the FDA. So what can we do? If any of this kind of spurs you to want to kind of maybe act a bit, or at least kind of behave a bit differently in practice, it did kind of change the way I approached drugs after our kind of project. And again, I've been in academia for 15 years prior to that. I think one, just being aware of this is a great start. So I learned a lot when I went through this process. Things I probably should have known through my training, I didn't know, and just understanding the process a bit better has helped me in practice in 2016. So know that the package insert and even the published literature may not give you the complete story about that drug. Look for the clinicaltrials.gov label on articles. So all the major journals decided about a decade ago that to be published in their journal, you need to be registered at clinicaltrials.gov. I can tell you that doesn't happen a lot of the time. Even in the major journals, that often is not occurring. But looking for that kind of, was this trial pre-registered, I think is helpful. Let journals know if it wasn't pre-registered, and email them, ask them why this wasn't registered. So, number 3, be leery of small trials published in smaller journals. So I think really those smaller trials may just represent a tip of the iceberg in that area, and there may be many other trials that are not published that had opposite findings. So I consider those trials to be hypothesis forming and certainly not something to change my practice. And last, don't be afraid to be heard. It's pretty easy to get your voice heard at the FDA. FDA.gov has easy ways to communicate with the FDA. It is our taxpayer dollars, and if you think something should be changed, they actually don't mind hearing from you. So I email the FDA several times a year and have yet to not hear back. You always get something back from someone at the FDA responding to your thought or criticism. So if you come across a trial or something in the label you think is kind of odd, don't be afraid to let them know that, and chances are you'll get a response that might start a productive dialogue. So again, a lot that I learned in this process. Some of this you guys may have known. Hopefully some of it was new. And we have a few minutes, and I am happy to take questions for as long as you guys would like. And thanks again for the invitation to come across this side of the Willamette. So.
I would like your comment about marijuana.
Okay, that's off topic, but sure. And what's that? Yeah, the question was about marijuana. Is there a more specific question than just my opinion, American?
Is it beneficial for certain conditions?
Yeah, I would say yes. So without going into great detail, I think there were good reasons for medical approval of marijuana. I don't mind telling you, I did not vote for legalized marijuana when it came up on the Oregon ballot a couple of years ago. I'm not sure Oregonians need to be smoking any more marijuana than they already are, so I did not vote for the public initiative. But yes, I think medical marijuana does have some legitimate use. So we definitely use it in our oncology population. So being here in the cancer center, I imagine there's some clinicians who would feel strongly about, and some of their oncology patients, it can be a wonderful antiemetic. For general pain, it's not, I don't do much with marijuana, and I would not broadly advocate marijuana beyond the couple areas where it's been shown to be effective in literature, which is mostly in your guys' oncology population.
Does it affect driving?
Yes, yeah, you definitely makes you impaired. And there's a couple of PhDs at Oregon State who are involved with trying to come up with a testing system that the police could use when they pull you over. But yeah, no one here has to confess if you smoke marijuana in college. But for any of those who might have, no, it does kind of impair mental faculty. And yeah, I don't want someone getting behind the wheel depending on how much marijuana is in their system. So there probably are safe levels and unsafe levels. And similar to blood alcohol testing, there'd probably be some measure of marijuana testing to know at what point you were safe or unsafe, for example, to operate a motor vehicle. But yeah, at some point you were unsafe. So, which I won't say I know personally, but yeah, from the literature. Yes?
In some of these studies, like with this Zoloft and panic attacks, that they have just borderline statistical significance, but the reviewers think this doesn't cut it.
Right.
Can they put their opinion, or are they legally obligated once it shows some statistical benefit? Are they legally obligated to make an approval?
Yeah. No, it's interesting. So if you have your, like for panic disorder, for depression, for SSRIs, if we kind of stick with that example, because every drug is a bit different, but in that area, if you have two trials that are statistically significant, in the opinion of the reviewer, you're pretty much set for your indication. And when I say in the opinion of the reviewer, they can use a preponderance of, for example, secondary efficacy data to say they think this works, to label it a positive trial. So it could be that the main objective, which might be % reduction in panic attacks, that might not get significant. That might not be significant. Maybe the p-value was 0.1 for overall reduction in panic attacks. But they're going to have several secondary endpoints they're measuring through that trial. And the reviewer could say, based on the preponderance of data, these other outcomes, I'm going to call this a positive trial. That counts as one of two positive trials. So yeah, you could have a negative p-value for your primary outcome, but you got to have other stuff that's positive. You can't be a completely negative trial and have the reviewer say, I'm going to call this positive. So they are doing their best, but it is a bit of a flawed system in my opinion.
Can the manufacturer then come back and sue them or something?
No, can't sue the FDA. So you can object, and there's kind of processes for appeal, but no, not in that kind of sense. So, John.
That was great. I think I'm interested to hear what you think about your proposal on who should review it. It's kind of hard for me to understand why people would argue against having all the data transparent and putting it out there. From my own limited experience with the FDA and some meetings about the LAVA controversy, I was struck at how there's a very small group of people in each wing of the FDA, like the respiratory group, and the personalities run the show. So I'm not sure I think the FDA is the group to do what you're proposing, but maybe someone like HRQ or some of the other government agencies that are focused on cost effectiveness, et cetera. But
Right.
I don't think the FDA would be my first choice to tell me whether a drug's cost effective or efficient. But I don't know what your thoughts are.
Yeah. So sitting to that kind I think the FDA now is probably pretty well positioned to tell us, like, a little more quantitatively how effective is this drug. So, I mean, this kind of our area we looked at is a good example of where there is quantitative data in those packets that are just nowhere in the labeling. And it would not be hard to add an extra line to labeling for Zoloft, for example, and tell us what was the % reduction in panic attacks for the drug compared to placebo. And then leave that up to us as clinicians to decide, all right, this is going to affect me in the way I prescribe. I tell you, the argument against that from like some of the family medicine attendings when I presented this is they kind of say, you know, if this drug works, that's all I need to know. That's, you know, I want to know this is an option for panic attacks. It has multiple trials showing it works. That they weren't that concerned that the placebo effect was 45%, the drug on average was 60%, and that's not much of a difference. That whether or not the placebo was 10% and drug was 80%, they said, you know, if it's available, I might use it, and knowing that wouldn't affect it. But I would agree with your kind of main point. Maybe the FDA is not the best to say, this drug is cost effective, or among these 6 ARBs or ACEs, here's the one you should use. And they, at some level, they can't do that, because we all get different prices for different drugs. And we have too complex of a medical and health insurance system in this country for the FDA to say this is the most cost-effective drug. But what they could do is provide more of that quantitative data, or to make sure that if they have it, it's getting into the public realm, that we're not dependent on the manufacturers to decide what's going to be published for us to see. So that when I do a literature review for antidepressants, I should be able to get an accurate view of what the data actually says, and not a skewed view based on what the FDA has versus what's in the public realm. And the FDA could do things to fix that, to either determine kind of anything we have has to move forward into the public realm, or to put in the package insert that quantitative data. But I agree, they would probably not be the agency to then say, based on this quantitative effects, here's the best drug. But I think we'd all be better positioned that to the extent that we're involved in guideline committees or with things like the American Thoracic Society, American Diabetes Association. It's sometimes hard to get that. You know, we get what's in the published literature. But again, that may not always be all you need to know to kind of make the best opinion. And if you are helping to write guidelines that are seen by insurers and patients, and you'd like to know I've got all the data. And I can tell you, this was, you know, I kind of joke, we had a postdoc from the Netherlands who did a lot of the kind of backbreaking work on this paper. I mean, it was really hundreds of hours of time with these cumbersome PDF documents from the FDA to basically learn, hey, the drug's not as effective in the FDA's view as it is in the published literature view. So, and I think relying on a postdoc from overseas to come do hundreds of hours of work so we as prescribers can figure out this drug's not as effective in the real world as it is in published literature, you know, that there probably needs to be a better system than that for that information to get disseminated. So I don't have an easy fix for you, but it's certainly, I think, a topic that deserves ongoing debate as it's getting. Yeah.
Thank you very much.
Yeah.
One of the quandaries we have is we have drugs being developed that we don't really understand the mechanism of action and the mechanism of the disease and where actually the drug, where we need to actually target our interventions.
Yeah.
I really like the idea of putting out the information around the magnitude of effect, but I would also like to challenge us, I guess, all of us, that we really need to understand the persistence of effect. Some of these drugs that people are like Zoloft or diabetes drugs, they're going on for a long time. Do we know that they really work over time? And is there a drop-off or a change? And so I'd like you to hear what you have about that.
No, yeah, so the comments around, you know, and I think we could phrase it as a very broad question around, are we using the right endpoints in these trials for a drug to get approved? And as Liz alluded to at the beginning, that's a controversial topic now in general. So in the diabetes world, there's been a bit of an upheaval around how does a drug get to market and what do we approve it based on. But yeah, I agree with you. In the area of psychiatry, which I think is a particularly challenging area for drug approval, because it is wonderful we know what a drug does. And we now know where metformin works in the liver. I know what it does for diabetes. I don't know what exactly Zoloft's doing in all the different parts of the brain that may contribute to anxiety and or depression in my patient. So for drugs in the psychiatric world, it's very difficult to link mechanisms to kind of efficacy. But you certainly can still ask the question around, is a 12-week panic attack study the best thing to say, here, prescribers, is a drug, go use it in your patients for 6 months or 6 years or as long as you want. And there's editorializing on that as well. Should an endpoint be a 1-year outcome study where the endpoint is more who's still at work? Who's got productive relationships with family members? And yeah, you can start asking some very broad questions around, are these appropriate endpoints? But it gets prickly pretty fast. We would lose an awful lot of drugs from the market if we start setting thresholds around things like total mortality for diabetes drugs or long-term public health for psychiatric drugs. So, but I think there is a robust debate to have there. Those are things you can communicate with the FDA on. Looking at the labeling, you say, all I know is 12-week data. What do we know about longer term? What happens at 1 year or at 2 years? And sometimes there's some data out there on that. That phase 4 I alluded to at the beginning, you do have to do phase 4 monitoring of drugs on the market. But that does not necessarily mean efficacy. That basically means monitoring for is something bad happening that we need to know about. But you do sometimes get 10-year or even now 15-year efficacy data from some of these trials. They'll keep following patients over time, and you'll get sometimes decade-long results published. So some of that is getting out there now. But yeah, it does beg the broader question of just, are we always using the right endpoint for drugs in these studies? And that's where I think a discussion between prescribers and the FDA can be very effective. And as John alluded to, you can even get involved with the FDA. There are clinicians on the review committees that make these sort of determinations for these drugs. So that's all public knowledge. You can get the minutes from those meetings, and you can dialogue with those reviewers and see who they are. And so, but yeah, it does take, I think, an active, involved prescribing community to be part of the solution to this. Yeah, so please, hang around if you want to come up. So thank you again. Thanks for the invitation.